Welcome to the Strategic Considerations for a Successful CNS Clinical Development Pathway conference call. My name is Carol, and I will be your operator for today's call. At this time, all participants are in a listen-only mode. Later, we will conduct a question-and-answer session. During the question-and-answer session, if you have a question, please press star then 1 on your touch-tone phone. Please note that this conference is being recorded. I will now turn the call over to Beatrice Setnik, Chief Scientific Officer at Altasciences.
Hello, and thank you for joining today's session. And I do hope that everyone out there is keeping safe amongst the COVID-19 pandemic. And I will today be bringing the science home remotely to you. And we will be talking about some of the considerations that one needs to take when developing a CNS-active compound. The preparation of any drug development program is really half the battle, but being able to strategically determine what type of studies you may need, what type of questions regulatory bodies will be asking, and helping you navigate to not only a successful regulatory submission, but also whatever key milestones you have in development, whether it be licensing opportunities or other investment potential. We want to help you determine what would be the best strategy for your CNS active compound and being able to provide compelling data. Very briefly, I'd just like to introduce you for a moment to Altasciences. We are a clinical research organization that is a mid-sized full-service CRO with over 25 years of experience. We do have specialty experience in CNS drug research, including abuse and dependency evaluation, which is going to be a lot of the top focal point topics for discussion today, with locations both in the US and Canada. Our state-of-the-art facilities include controlled smoking and vaping chambers, and we are the only CRO within North America that has permanent driving simulators. Also, just to give you an overview, we have a preclinical facility in Seattle, as well as clinical sites in both Kansas and in Montreal, spanning North America. We have bioanalytical services both in Canada as well as the United States, and have recently added a manufacturing service site. For today's session, we will be learning how to leverage your early phase clinical trials to capture relevant data in order to characterize your CNS active drug. We'll also be looking at how to determine if you need additional studies that may accurately project your clinical development timeline and have more informed discussions with regulatory agencies. So we'll be taking a look at different perspectives of drug development. As a way of introduction, CNS active drugs, and that also includes cannabinoids, have unique attributes that are distinct from other types of drugs in development. When you're developing a CNS active drug, you also need to take into account the cognitive impairing or enhancing effects the drug may have. Particularly from a safety point of view, one has to look at the potential reinforcing effects or the abuse potential. Is the drug likely to become abused once it becomes marketed? Does the drug have physical dependency and tolerance concerns that may affect patients and both patients and non-patients? And does the drug have additive effects? So for example, when it's combined with drugs, other drugs, or with alcohol, can it cause more impairing or some other safety issues? If some of these may be relevant, particularly abuse potential, there may be a need for that the drug may need to be scheduled or controlled under the Controlled Substances Act. And this becomes very important in determining what is the likelihood that your drug may need to be scheduled, as scheduling does have an impact on availability, on prescription refills, and other nature of drug accessibility, as we will discuss later in today's session. And it's imperative to know from the onset when these additional studies may be needed and at what point will you be making pivotal decisions as to whether or not you may need to do additional testing. There are several regulatory guidances from the FDA that will help you along the pathway of CNS drug development. And these span a range of topics. They include the abuse potential, driving simulator studies, suicidality assessment, as well as specific guidances for botanicals. The timing and review of some of these meetings as you course throughout your drug development trajectory you will have various opportunities to speak and interact with the FDA. When it comes down to some of these specialty safety studies like abuse potential, many of these discussions don't occur until about your end of phase 2 meeting. Now, the information and data that you collect prior to the end of phase 2 becomes critical in determining at that meeting whether or not you will need to do additional studies for abuse potential, driving simulator, or any other studies that may have not been included thus far in your program. Being aware that these studies may be a possibility is important, and being able to review your data ahead of time may also give you enough evidence to present that you may require or request a waiver to do these studies. What you don't want to do is get to your pre-NDA stage and determine only then when the regulatory agencies flag that you may be missing critical data. And that might be a discussion that may actually hold up your NDA, which we've seen sometimes happen with sponsors, or it may prompt then a post-marketing approval requirement. So it is important to really have a very good, well-defined strategy as you go into an end of phase 2 meeting and really have these points strategically lined up ahead of time. When you're preparing for an end of phase 2 meeting, for example, some of the critical areas that you'll be thinking about as you move towards, and these areas really start from the very onset of drug development. These are things that should be relevant for consideration from the very first time that the data on the drug is being collected, even in the preclinical phases. Does this drug show impairing effects? Does the drug bind to receptors or targets that are already associated with abuse potential? Is it similar to drugs with known abuse potential? Does it have a pharmacological or adverse event profile indicative of abuse potential? And does the drug have the potential to cause physical dependency? Some of these questions can be answered in your very early preclinical work as well. So sometimes you'll already have indicators as to whether or not there may be evidence that these additional clinical studies may be needed down the road. The FDA recommends looking at various different aspects of the clinical pharmacology of a drug. And in particular, during your clinical development program, there will be triggering adverse events that the agency will be particularly interested in during your end of phase 2 meeting in determining whether or not you may need to do additional studies for safety. And they're looking for different terms related to euphoria, for example, euphoric mood, feeling abnormal, feeling drunk. But there are also other types of adverse events related to psychosis, for example, drug tolerance, withdrawal symptom. This list is not definitive, but it is a relevant guide to steer you into the direction of looking at your data and evaluating it as to whether or not further work may be needed in the areas of abuse potential or withdrawal dependence evaluation. Being able, in some cases, drugs may need to undergo testing. In some cases, sponsors may be developing drugs that have actually less abuse potential or have a safer profile compared to already marketed drugs. For example, developing an analgesic that is non-opioid related may give you a completely different profile that with a compound that may not necessarily have abuse potential. In some cases, when you do have a more promising safety profile or better efficacy compared to other marketed products, it may give you the benefit of being able to request priority reviews by the FDA. So fast track designation, for example, and understanding and collecting that data early on in your development program can help you get priority reviews with the FDA, which will facilitate your drug development process, and that may be quite advantageous. So now I will move on to the first set of specialty studies that may or may not be requested by the agency, but it's good to know that they may be required and that these may need to be addressed at an end of phase 2 type meeting. And these are the driving simulator studies. There is a specific guidance on this, and it's important to understand that this is an important safety and public health priority, understanding if a drug impairs the ability to operate heavy machinery, including motor vehicles. The FDA recognizes that patient self-perception is not adequate for determining the degree of impairment that a drug may have, and therefore there are more objective ways of determining whether or not a drug has an impact on driving and those types of psychomotor skills that are required, which is a complex battery of skills required to pay attention, to operate to have the psychomotor skills to hold a vehicle in a position. And so these are there are dedicated studies that will look at these elements. Which types of drugs need to be evaluated? Well, typically for driving simulator studies, you're looking at drugs that are intended for chronic or chronic intermittent use by adults who drive. This would include both psychoactive and, remember, non-psychoactive drugs may also fall under this category. So, for example, if someone has impaired consciousness from a hypoglycemic reaction from a glucose-lowering drug or has impairments in vision, for example, some mydriatic drugs, those may also be warranted to do further testing in a driving simulator mode. So it's not always necessarily CNS drugs. If they have the potential to improve driving performance by decreasing somnolence but might increase risk-taking, for example, stimulants that may result in aggressive driving, those also may be subject to further testing. And oftentimes drugs intended to be taken at night, which may have residual daytime effects like grogginess, can also be subject to these types of studies. So if you have a drug that may be need these type of evaluations, if you're looking at a different indication, even for a marketed drug, if you're looking at a different indication, a different dose, different dosing schedule, or if you're targeting a new population where there's insufficient information about driving, even marketed drugs that are repurposed or reformulated may require additional testing. This is just an example of the Ambien label, which has dedicated information in its prescribing information regarding to driving simulation. In this case of Ambien, you have complex behaviors such as sleep driving that are possible, where people will go off and drive and not be consciously aware and don't remember the fact that they drove. In the section 5.5 of their label, they do caution against patients engaging in any types of hazardous occupations or anything including operating heavy machinery or driving a motor vehicle after ingestion. So oftentimes this type of data will make its way into a product's label if there is important safety information to relay. The FDA endorses a tiered approach to assessing driving impairment. And so even though oftentimes these studies are done later in the program, in the drug development phases, the preliminary investigation into different impairing effects and effects on cognition are done very early on in the program. And that will help you determine whether or not you may need to do these later in your clinical development program. So even as early as first-in-human studies can incorporate measures of different arousal, wakefulness, attention, processing speed, psychomotor functions in more standard, smaller batteries that enable you to determine whether or not your drug does cause any impairing effects or perhaps cognitive enhancing effects, and whether or not future studies may be needed. If you collect sufficient information to show that you don't have impairing effects, you may in fact leverage that data to request the fact that these types of studies may not be needed in your development program, and that of course helps to fast track your program. The early testing can start with first-in-human trials, and some of the advantages of conducting these types of CNS analyses early on is the fact that early trials include higher doses. It allows you to characterize pharmacology better. It also allows you to do some very good correlations between the subjective-objective effects and pharmacokinetics because you have intense PK sampling early on in your development program. And it also helps to collect important safety information that you can then carry on for your patient trials when they're taking medication home in phase 2, phase 3, and determine what may be important safety parameters to include in those protocols. So there are a number of reasons why it's important to evaluate the CNS effects early on in a clinical development program. Later on in phase 2, phase 3 trials, the assessment of CNS drug effects continues. When there are drugs with potential impairing effects, the FDA recommends that PK assessments be also included. Of course, these may be more sporadic collections in subsets of the population, but it does allow you to relate impairing effects or CNS effects to exposure. One should also look at different factors that can affect exposure, including food, time of dosing, concomitant medications as that becomes more prevalent in patient populations as you move to phase 2, phase 3. You're also looking at monitoring specified intervals for temporal impairment of risk. So oftentimes looking at open-ended and targeted adverse event collection. You certainly want to be reporting any kind of reports of motor vehicle accidents or traffic violations, perhaps during the course of phase 2, phase 3 trials. And any types of effects that haven't been previously identified is the time during phase 2, phase 3 to take a look at those. For example, impaired executive function or memory or other types of cognitive domains. Dedicated driving studies are done in the cases where there may be a need to do so. So if there is collective evidence that a drug may have an effect on driving ability, usually dedicated tests and studies are needed, which requires one type of study design. And these can be done with actual motor vehicles, or more frequently in North America and accepted by the FDA are driving simulators, which are very realistic scenarios as shown here in the picture. And these are looking at the different domains required for a complex task like driving a motor vehicle. What's pictured here is the CRCDS mini-sim. It's a simulator. It has a very realistic simulation of the cockpit in a car. It meets the NHTSA standards. And it employs all of the kind of driving experience that one would if they were sitting in a real car, with the ability for a tester to be present in the room. And of course, there's no safety concern over navigating a vehicle under impaired conditions in this case, because it's done in the safety of a clinic. When we're looking at some of the methodological considerations for driving studies, the FDA recommends to enroll patients who are from the population likely to use the drug, but that is usually done in healthy volunteers unless there's a notable difference between the patient population and the healthy volunteer population in terms of cognitive effects. The doses, you look at different, evaluate the effects of initial dosing and the effects after chronic exposure, so oftentimes it can be done after a few doses. Testing can be done at maximal levels of parent and or active metabolites. So you're looking at usually the highest exposures to be encountered during clinical use. We also include a positive control, and then that enables us to demonstrate assay sensitivity and understand the magnitude and duration of impairment. The time of day and duration of effects should be included and documented, as well as tested. So for example, if you have a drug that is taken at night and may have residual daytime effects, you certainly want to replicate that clinical scenario and test as well in the morning, the day after, for those types of drugs. Crossover designs are typically employed because these are smaller numbers. Subjects act as their own controls. And we're looking at very carefully nighttime and daytime dosing, onset of duration, the extent of the duration of the exposure, and how that affects the ability for a person to drive. As we see from different published studies, there's a wide array of sensitivity of driving simulation across different various compounds. So for example, flibanserin actually has showed a better performance compared to placebo when you're looking at standard deviation of lateral position. So this is deviation from the center of the road when you're driving. And of course, you want to be as centered as possible when you're driving. As you get down this list, you can see that other types of drugs like alcohol and some of the benzodiazepines cause more and more of a impairment on the driving ability as one would expect with this class of medication. In the endpoint analysis at the end, you're looking at examining the mean effect difference from placebo, and you're also comparing it to the active control. There's usually a responder analysis that's looked at to see if there are any meaningful impairments in the analysis across the different measures that are being taken. And we're looking at the relationship between the drug concentration and the different driving endpoints that are collected throughout these types of studies. And those are some of the critical studies, and like I've shown with the Ambien example, that may be included in the label. So now we will go over to another element of CNS drug evaluation that's very important and unique to CNS drugs, and that is the human abuse potential evaluation. And this is the type of data and study that's collected if there may be signals that are indicating that a drug may have abuse potential. Now, some sponsors, if they're developing a drug that is unique and is actually maybe suspected of having a far safer profile with respect to abuse or misuse, some sponsors choose to conduct these studies as a strategic way to demonstrate decreased abuse potential and to differentiate from standard treatment. So, for example, an analgesic that's not acting through a new opioid pathway may decide to differentiate itself based on the currently approved Schedule II opioids. So just very briefly, the Controlled Substances Act, enacted in 1970, is essentially a method of controlling substances that have a currently accepted medical use in the United States, but have a relative potential for causing abuse or dependence. And just to differentiate quickly, physical dependence is a neuroadaptive process that can occur in the absence of abuse potential. So, for example, drugs like antidepressants can cause you to become physically dependent, meaning that if you suddenly withdraw from the medication, you may exhibit withdrawal symptoms. This often occurs with drugs of abuse, but it's not always necessarily paired with abuse itself. And we'll go into those examples just in a few minutes. So the scheduling is actually done at the time of the NDA review. And it's important because once your drug is scheduled, it's very difficult to get a less conservative schedule over time. In fact, most drugs move from a less conservative to a more conservative schedule, with sometimes in cases where post-marketing data is collected to suggest that there may be more abuse potential than was originally thought at the time of scheduling. So it's important to get the relevant data concise and be able to justify whether or not your drug may or may not need to be scheduled, and if so, what would be the appropriate schedule it should fall under. This is, for example, the 5 classes of scheduling in the United States, going, as you see from the left-hand side, Schedule 1 to 5. As you go down the list, there is less and less abuse potential. The main difference between Schedule 1 and 2, both have high abuse potential. Schedule 1 has no accepted medical use. It's for research purposes only, whereas Schedule 2 has accepted medical use. So, for example, Schedule 1 still includes marijuana because it's not federally legalized, so it still remains as a not accepted medical use in the United States, whereas the oxycodones and the different opioids are in Schedule 2, for example. And then as you go as you decrease from Schedule 2 to 5, you'll see here in the middle column that you have more and more loose restrictions around the prescribing practices, to the point where a Schedule 5 could potentially be over the counter. But a Schedule 2 has very strict, there's no telephone prescriptions and no refills allowed on those. So certainly scheduling will impact the accessibility to a medication. So it's an important element to consider in the CNS drug development process. I want to give you an example of a compound, dronabinol, that had differential scheduling based on formulation differences. So, for example, Syndrose was approved in 2016 as a Schedule II substance. It was formulated as an oral solution, and what was found during the review was that the Schedule II was given as a conservative scheduling due to the formulation, the fact that it's an oral solution. It was deemed that it could be easily manipulated for abuse by inhalation and other routes. So just so that you know, dronabinol is related to THC. It's easily evaporated. It contains alcohol. And because of the parameters of this formulation and the thought that it could be easily abused and lend itself to abuse, this received a Schedule II status. The other points here, and I just wanted to also mention that other types of dronabinols like Marinol, for example, THCs, were actually Scheduled 3 at the time. And that was rescheduled to 3 in 1999 to increase patient access. Even though it's the same as Syndrose, it has a different formulation, so it was deemed less potentially lend itself to abuse as easily as Syndrose would. So there are key differences if you're if you're targeting an FDA approval versus an EMA with respect to abuse potential. In Europe, the European agencies primarily focus on preclinical studies for the evaluation of abuse potential, whereas the FDA and Health Canada focus not only on preclinical but also on clinical evaluation. And that's an important differentiator to know, particularly if you're planning on filing in the United States. So abuse potential is assessed for any new drug, and it can also be assessed for marketed drugs. So if the drug affects the CNS, if it's chemically or pharmacologically similar to drugs with known abuse potential, or if it's a novel drug, even in cases where the mechanism of action may not be well characterized, but it produces certain psychoactive effects that may be indicative of abuse potential, there's a high likelihood that that drug may need to be evaluated for abuse potential, and then a scheduling determination be implemented at the time prior to approval or at the time of approval. Even for marketed products, the same way as driving simulators, if a product has a different formulation or a different target or a different formulation that may lend itself to increased abuse, it may also need to be reevaluated for abuse potential. So again, in terms of your development process, these conversations are usually had with the FDA at the end of the phase 2 meeting. But prior to that, you've already collected very critical information that the agency will review to determine whether or not you will need such a study. And understanding that information and orchestrating and pulling it together in a meaningful way in your briefing documents and your presentation at the end of phase 2 meetings strategically can help you really differentiate as to whether or not you may need that study and what the rest of your clinical development program, your budget, your timelines may look like if you don't, whether or not you may need to do these types of studies. So they have a potential big impact on your program, and it's very important to identify whether or not you may need to do those later on in development. And in fact, if you have a very good profile, it may be very advantageous for either fast-track designations or for licensing or other opportunities to show that you have a distinct safety profile that may be unique from other marketed drugs in your therapeutic area. The human abuse potential study, if it's needed, is a single phase 1 type study. It evaluates the likability of a drug in a face-valid population. So these are typically conducted as single-dose, double-blind crossover studies or single-center. They're conducted in a face-valid population, which are non-dependent recreational drug users. So these are people that are not physically dependent. So when they come into the clinic, they're not going to have the craving and the withdrawal symptoms that would be problematic to medically manage. They're non-dependent, so they're the kind of sporadic recreational drug user. In these studies, because most of what we're asking are subjective questions, these are both placebo and active controlled in a very robust double-blinding process. Active controls are scheduled controls with known abuse potential, and those can be classes of opioids, stimulants, depressants, and others based on what's appropriate in comparison to your pharmaceutical drug. The pharmacological challenge is used as a mechanism to ensure that subjects are, first of all, non-dependent and that they're actually sensitive to the active control. So even though they're reporting that they have recreational drug use, you still want to pharmacologically validate that to ensure that they, in fact, can tell the difference between your active control, for example, morphine or oxycodone versus placebo. And that's based on their tolerability and their ability to respond to subjective measures in an appropriate way to differentiate the two. And that's usually always predefined in the protocol. In some cases, and many cannabis products are coming out, particularly in Canada where it's now federally legalized, there are studies that require evaluation of abuse potential, but require additional parameters to look at different routes, including smoking, inhalation, vaping. So we have controlled smoking rooms in order to be able to safely administer the vaped or smoked or inhaled product in a setting that also minimizes the exposure to staff. so the safety has to be always first and foremost. The human abuse potential study design includes a variety of different subjective and objective measures. Typically, the primary endpoint is drug liking. So as you can see from this screen, it's presented on a 0 to 100 point visual analog scale using computerized batteries of tests that the subjects have a lot of training on beforehand to ensure comprehension and ability to use the computerized modules. The analysis of data is based not on a single endpoint, but really a whole collection of endpoints and the totality of the data to demonstrate the abuse potential. This is some data, for example, from Epidiolex that was recently approved for various epileptic disorders in children and adults age 2 and over. This is showing you drug liking. It's a bipolar scale, meaning that neutral is in the middle at 50. Strong disliking is towards 0, and strong liking is towards 100. And these are the different controls and active ingredients that were administered over a 12-hour time course. And placebo is here in orange, and as you can see, dronabinol and alprazolam in gray and pink here in red were the active controls, and these showed substantial drug liking on the scale, whereas the cannabidiol showed very similar profiles that were very close to placebo. So it didn't show the magnitude of abuse potential compared to the active controls. And as such, Epidiolex received a very low schedule of Schedule 5. And the wording in Section 9.2 is really stating that it produced responses on measures that were within the acceptable placebo range. So it really does emphasize that there really isn't much abuse potential with this particular compound as it's worded in its labeling. At the time of scheduling, when all of this data is submitted, as I said, the human abuse potential study is just but one of the studies that supplement data to make an informed scheduling decision. The agencies that are involved in the review of your data and the decision-making for scheduling involve not only the FDA, but NIH gets involved, as well as the DEA, which ultimately makes the ruling on the drug schedule at the time of approval. Sometimes sponsors are waiting a few months after approval before the scheduling status comes in. So I've seen that happen as well. But everything collectively gets reviewed. So the chemistry studies, receptor ligand binding studies, PK studies in both animals and humans. There are dedicated abuse studies that are conducted in the preclinical setting, and that includes general behavioral pharmacology, drug discrimination, self-administration. There's a physical dependency study that's conducted in animals. And there's also the human studies, the human abuse potential study, which I just spoke about, physical dependency studies, which we'll get into in just a moment, and your abuse-related adverse events that I had spoken a little bit earlier about are examined from your entire clinical program. So all of the clinical studies that you've conducted to date, as part of your NDA filing, there will be an examination of those signals and abuse-related events across the board. So it's really looking at the totality of the data. The human abuse potential study is just one fragment of all of the data that's reviewed collectively to determine drug safety and ultimately drug scheduling. So it's important to keep an eye on how your drug is, what kind of data you're producing, and periodically review it throughout your drug development program, because it will be very predictive for you as to what will be needed and ultimately what kind of scheduling may be granted for your drug. The methods that the controlled substances staff use to determine scheduling is what's called an 8-factor analysis. And usually they organize the data in the 8 factors that are shown here to determine, to evaluate all of the data that's submitted and determine and to present a suggested schedule to the DEA for ultimate ruling. One of the proactive and strategic things to do as a sponsor, because you are required to put a scheduling recommendation in your NDA, is to prepare the 8-factor analysis in a very much similar way, and for you to have an opportunity to look through that data and make a meaningful suggestion, proposal in the NDA as to whether or not the drug should or shouldn't be scheduled, and if you feel it should be scheduled, what schedule it should fall under. So it's a more of a proactive strategic approach to the NDA filing, and this is something that, of course, we can help with at the time when you're ready for these types of submissions. Now I will jump into another aspect of abuse potential, but more towards the physical dependency side. And this is one that often gets overlooked, and it's very important. And understanding what the requirements around this are can really prevent you from having costly delays in your NDA submissions and ultimately in approvals. So I'd like to spend a few minutes particularly on this topic. The physical dependency, like I said, it manifests because your body over time will adapt to a drug. And when that drug is suddenly discontinued, or when there's a rapid dose reduction, or you administer an antagonist, for example, it can cause often intense withdrawal syndromes. And this is an important safety element that needs to be defined during a clinical development program. And the guidance from 2017 that was issued on abuse potential has sections specific to dependence. And you'll see that there's information on dependence in the prescribing information of drugs. There are various symptoms that range from mild to serious. Some of them are general CNS that occur widely with CNS drugs, and those things can include things like nausea and headaches and agitation and sleep disturbances. And some types of withdrawal symptoms are very specific to certain CNS drugs. So, for example, the lacrimation, the rhinorrhea, and sneezing that you see with opioid withdrawal. For SSRIs, for example, you can have oftentimes electric shock sensations, confusion, myoclonus, and those are, and I just want to point out here that SSRIs can cause quite pronounced withdrawal symptoms, but SSRIs are not abused. So that's an example of a drug that may have physical dependence potential, but it does not have abuse potential. But nonetheless, the safety considerations for withdrawal of SSRIs are very important to evaluate during your development program, so that meaningful information, if there are any safety concerns, can go into the prescribing information. Usually the withdrawal symptoms are transient and reversible, but otherwise sometimes they can be major complications from abrupt drug discontinuations, and those can be things like seizures, suicidality, psychosis, that can be of serious consequences, and therefore these become very important to evaluate within the context of a drug development program. The importance of this safety information is recognized by regulatory agencies, and it's important that this data is available to inform physicians and patients and drug abusers about the consequences of abruptly discontinuing medication, whether or not there's a potential need to examine tapering or switching to alternate drugs with similar mechanisms of actions in cases where patients are being weaned from a drug or taken off of drugs. What to do to medically manage patients that suddenly have withdrawal symptoms, how to identify it, how to relate it to a drug withdrawal symptom, and how to medically manage it. Very important clinical considerations and important data to be collecting in the course of a drug development program to have a better informed label for safety reasons. There are different types of drug discontinuation effects that have various clinical manifestations. There are what are the acute withdrawal symptoms. So these are the signs that come very quickly on after a drug has been discontinued. So oftentimes diarrhea, GI cramping if you've been withdrawn from an opioid. There also is a rebound effect, which is a recurrence of symptoms of the treated disorder in patients that's usually more severe than before the treatment. So for example, severe depression after the discontinuation of an SSRI that was even worse so, or anxiety after the discontinuation of a benzodiazepine. Then there's also post-acute withdrawal disorders, and that's the appearance of symptoms relating to emerging new disorders, like, for example, tardive dyskinesia or protracted insomnia. So there's a variety of things that can happen even beyond the acute withdrawal symptom phase. The drugs that are known to cause withdrawal include both scheduled and unscheduled drugs. So like I said before, it doesn't always have to be with drugs that have abuse potential. Oftentimes it will go hand in hand, but it's not necessarily the case. So, of course, all of our usual suspects, the opioids, the benzos, the cannabis, the stimulants, can cause withdrawal symptoms. But a lot of unscheduled drugs can do that as well. Antidepressants, anti-Parkinsonian drugs, antipsychotics, beta blockers, corticosteroids. So it's one has to also cast the net outside of the usual suspects. And I think that's where a lot of sponsors get stumbled, because they don't always think that that might be an important safety evaluation, since their drug may not necessarily be associated with abuse potential. They're two distinct phenomenon, and they're both important safety parameters, but they don't always coincide. The assessment of physical dependency can be done both on a preclinical basis in animals, and it also is done on a clinical. And this is the part that's often overlooked. And I have had scenarios when I've been helping sponsors that got stuck at the NDA stage and had to go back and do further testing before approval. And this is the last thing that you would want during your submission to the FDA. So a clinical study is typically conducted in phase 2 or 3 after you have a exposure of at least 30 days. So most often it's done phase 2, phase 3, but you need to proactively plan it as part of your protocol. So you have to have pre-planned assessments post-study drug discontinuation. You may need to consider blinding. You need to consider what kind of endpoints, withdrawal scales you want to proactively put into the protocol. It requires a bit of thought, because it does have to be put into a section. It's not enough to simply passively collect adverse events after discontinuation. There need to be actual assessments specific to withdrawal, and this oftentimes gets overlooked, but this is something that we can certainly help with should you have questions around this. If, for example, a population, if you're in an indication in which it's unsafe to discontinue patients off of a study medication, then you can't do this in a phase 2, phase 3 study. You have to do this in a dedicated study in healthy volunteers. And this would require, so for example, in patients with epilepsy or schizophrenia, they can't be safely discontinued off medication, and so therefore these studies are done in healthy volunteers. Under this paradigm, it requires at least a 30-day exposure of drug used clinically. It's placebo-controlled. There's a lot of different adverse and validated withdrawal questionnaires that are implemented. Subjects are housed during the brief period around withdrawal so that they can be clinically monitored. They're very intensive. A lot of information is collected. One of the drawbacks in the healthy volunteer population, of course, is that things like rebound effects can't be monitored, because the healthy volunteers don't have the pathology that the patient population would. So there's some limitation. But from a safety perspective, it allows you to look at some of the acute withdrawal symptoms that would be observed. So these are important studies that are frequently overlooked, and so this is important to keep in mind that such assessments may need to be done, irrelevant of whether or not your drug has abuse potential. The types of withdrawal scales, there are many validated withdrawal scales. They're specific to different drug classes, opioids, benzos, stimulants, cocaine, marijuana, SSRIs, and general checklists. So if you are dealing with a new chemical entity that has a unique or undefined mechanism of action, oftentimes based on the pharmacology, you may need to incorporate more than one scale. Okay? Dependence does come into play into drug labels. So for example, with Epidiolex, they didn't have any symptoms or signs of withdrawal. And so therefore in their section 9.3, it says that this cannabidiol 1,500 milligrams per day did not produce signs or symptoms of withdrawal over a 6-week assessment period, beginning 3 days after drug discontinuation. So you can see here that the assessment period follow-up is substantial. So this needs to be really proactively planned in your clinical trials if you're looking at this. On the other hand, drugs that do have dependence potential also will have this in section 9.3. And for example, the Oxecta label here shows the different symptoms of opioid withdrawal symptoms in case that it occurs. And it states that in general, opioids should not be abruptly discontinued. So just quickly, some additional considerations for a CNS drug development pathway. So we talked about 3 critical areas that can't be overlooked during the program. In some cases, they can be waived, but it's so integral to monitor data early on and incorporate some of these measures of CNS cognition impairing effects early on into your even first-in-human studies so that you can start to build a case about the drug safety and leverage it as a strategy for your program. Additional things to look at, and that these can be looked at very early on in phase 1, are different parameters of cognition and psychomotor function. Things like attention, memory, perceptual function, psychomotor speed, coordination. All of these parameters can give you a very good indication of what your drug pharmacology is like, what kind of effects it has, and you can start to begin to make a strategy around the safety of your drug as well as the potential need for other studies and what impact that might have on your drug development timeline and your cost budget. Suicidality, also an important element. Columbia Suicide Severity Rating Scales are often included in evaluations of drugs, and that begins very early on in phase 1. And there is a dedicated guidance for that, so that is certainly another parameter of safety that's important not to overlook in your development program plan. So overall, for recommendations, the CNS drug development requires a unique safety evaluation because of the cognitive function impairment and abuse potential that drugs may have. So it's important to recognize that early on and incorporate assessments early on so that you can gauge and strategically define your program and have more meaningful conversations with the FDA at your critical milestones in drug development. Including assessments early in drug development allows you to better characterize your drug at both therapeutic, low doses, therapeutic doses, and supratherapeutic doses, because you will need to do those doses eventually if you have to do dedicated studies. And understanding what these preclinical and clinical requirements are upfront and being able to ascertain whether those are needed during your development plan can avoid very costly delays of learning that too late at the time of your NDA approval or at the time of your NDA submission. So this is something that really needs to be thought through carefully throughout your program. And like I mentioned, the 8-factor analysis at the time when you are submitting the NDA, the 8-factor analysis can be very critical in helping you understand your data, present your data in a meaningful way, and support what you feel would be an appropriate scheduling status for your drug or unscheduled status. So again, it really takes a lot of forethought and planning and strategic thinking about how to leverage your product in cases where you may have an advantage because you have a much cleaner safety profile. There are strategic ways in which you collect data to demonstrate that, to be able to get fast-track designations with the FDA, better enhanced labeling, have more meaningful discussions with investors potentially, or grant applications. So again, there's a lot of different reasons for collecting that data early on. It gives you a much better data package and ability to leverage that data package. So I will stop here and will be happy to answer any questions that may come through. Okay, so I have a question here. Are preclinical studies for abuse potential needed if you've already conducted a human abuse potential study? And the answer is typically if you are conducting a human abuse potential study because the FDA has requested that you do a full human abuse potential evaluation for a new chemical entity, then yes, you will have to do the preclinical as part of the full assessment. So I think the only times where you can go in without a preclinical human abuse potential study is typically when you're conducting an abuse deterrent formulation. In these cases, there's a separate guidance for the abuse potential evaluation of a deterrent formulation, and those types of studies are really not designed to look at scheduling, but to determine if a deterrent formulation has deterrent properties. So in those cases, those are unique and can really only be evaluated in the clinical setting, and in those cases, only the human abuse potential study is required. But they do usually include both oftentimes intended and unintended routes of administration. In cases where it's not safe to do an unintended route of administration, like for example, if a formulation gels too much and can't be safely injected, you couldn't do that in a clinical setting. Then there's our in vitro laboratory tests that can characterize and collect data on the difficulty of injection, for example. And there's a lot of very advanced methodology that laboratories can do with that type of testing. So that would be one unique area. Another question I have here is that a company here is developing a non-opioid analgesic that does not work on opioid receptors, so it has a different mechanism of action. Do we still need to conduct human abuse potential studies? Well, I think, and as I've mentioned across the program here, the need, the regulatory need to conduct them is really based on pharmacology and what you've collected. So if in fact you have a mechanism that's outside, that's not implicated in abuse, like it doesn't hit the opioid receptors, and if your entire program has shown that it does not produce the CNS effect, sometimes it may be a peripherally acting compound, then from a regulatory perspective, you likely will not need to do that. Of course, the FDA will review it with you and determine that at the end of phase 2 meetings. However, you may wish to actually engage in some degree of testing. You may not necessarily do the full gamut of preclinical, clinical, but for example, a human abuse potential study could be used as a demonstration with actual data to show definitively that you don't have abuse potential, and therefore you have a advantage over competing products that are the standard of care for a given type of indication. So in these cases, they may be strategic studies, but they're not necessarily regulatory mandated. So that's a different component of it, where you're now talking about how to leverage this type of data in a strategic element for purposes other than perhaps just filling a checkbox for regulatory purposes. So those are other elements of what you may want to consider during the drug development program. Another question I have quickly is what kind of preclinical models are required for abuse and dependence potential evaluation? Well, there's several. The typical go-to models for preclinical are the self-administration and drug discrimination tests. Those two are typically required. Any kind of behavioral data with animals showing whether or not something may be causing sedation or other effects will be looked at, but those two are the typical models for abuse potential. There are also studies, dedicated preclinical animal studies for looking at physical dependence symptoms, if that changes locomotor activity to a certain extent. I mean, it's not definitive and as thorough as a human study would be, but there are preclinical studies for that as well. Another question I have is, can I provide an electronic copy of the presentation? Yes, that will be provided. This video session is being recorded, so if you didn't have a chance to hear the presentation in its entirety, or you have colleagues that were unable to attend today, this session will be recorded, and we're happy to share the link with you. If you do have other questions that come up, I'm more than happy to help answer even after the fact. I will share my contact information with you following this session with the link to the presentation and to the recorded presentation. If you have specific questions that you haven't been able to ask today, or if you have a specific question that you'd like to reach out in terms of something specific to your program and what you might want to consider strategically for your program, I'm also happy to do that under the guise we can put a CDA if needed, but I'm available if you'd like to reach out with any questions as to how you might be able to leverage this data and present your data in a strategic way to meet not only regulatory objectives, but also your own drug development objectives as well. So I'm happy to help on that regard as well. So we will share the slides, the recorded link, and my contact information with all of you. So at this point, there are no more further questions. So I would just very much like to thank the audience today for joining today's session. If you felt that this was informative and would like to participate in future sessions, please let us know. Please let us know what your thoughts on this were. With all of the scientific meetings, unfortunately, being canceled in the near future, we'd like to be able to still keep the scientific flow of dialogue and sharing of information going. So we will have other future events, including another one tomorrow with some interesting poster presentation from our bioanalytical group. And we can keep you informed on any interesting sessions we have in the coming future in an effort to share the science in these challenging times. Otherwise, I wish that you and your families stay safe and healthy during this time. And please feel free to reach out if there's anything I can do to help further answer your questions based on the work you're doing. So thank you so much for joining today. I don't know if Carol, if you have any parting thoughts or instructions now at the end.
Thank you, ladies and gentlemen. This concludes today's conference. Thank you for participating. You may now disconnect.